Welcome to Wildlife for Sleep, a place where ancient oceans breathe slowly, where shadows drift beneath the waves, and where stories are told, not to awaken the mind but to gently quiet it. If these facts help you relax or slowly drift into sleep, please like the video, subscribe to Wildlife for Sleep, and share your thoughts in the comments. Your support helps this channel continue creating peaceful, slow science-inspired sleep documentaries. Now settle into a comfortable position, let your breathing soften, and allow the surface world to fade away. Tonight we descend into the deepest waters of the sea. Anglerfish belong to the order Lophiiformes, a diverse group of ray-finned fishes that includes more than 200 recognized species, distributed across both shallow coastal environments and the deepest regions of the open ocean. While shallow water relatives retain conventional fish anatomy, deep-sea anglerfish represent one of the most extreme examples of evolutionary specialization for life under high pressure. Near freezing temperatures and permanent darkness, fossil evidence and molecular studies suggest that ancestral anglerfish originated in shallower marine habitats millions of years ago, gradually expanding into deeper zones as ocean ecosystems diversified, as surface predators increased and competition intensified. Some lineages moved downward along continental slopes, adapting step by step to reduced light, limited food availability, and immense hydrostatic pressure. These migrations led to the development of traits that define modern deep-sea anglerfish. Life below approximately 200 meters enters the mesopelagic zone, where sunlight fades rapidly. Beyond 1,000 meters lies the bathypelagic zone, an environment characterized by complete darkness. Temperatures typically between 2 and 4 degrees Celsius, and pressures exceeding 100 atmospheres. It is within these layers that many anglerfish species have established permanent populations. Survival in this realm requires minimizing energy expenditure. Deep-sea anglerfish evolved soft bodies with reduced skeletal density, allowing neutral buoyancy without constant swimming. Their musculature is limited, reflecting an ambush-based hunting strategy rather than active pursuit. Fins are adapted for slow hovering and precise positioning in still water. Metabolic rates in deep-sea anglerfish are among the lowest recorded in teleost fishes. This slow metabolism enables individuals to survive weeks or even months between feeding events, a critical adaptation in ecosystems where prey density is extremely low. One of the defining innovations of anglerfish evolution is the elysium, a modified dorsal spine extending forward from the head. At its tip lies the esca, a specialized organ capable of producing light through symbiotic bioluminescent bacteria. This feature transformed the anglerfish into a highly efficient predator in darkness, allowing it to attract prey rather than search for it. As anglerfish populations settled deeper, sensory systems also changed. Eyes became highly sensitive to faint bioluminescent signals, while mechanoreceptors and chemoreceptors increased in importance. These adaptations allow anglerfish to detect vibrations, pressure changes, and dissolved chemical cues in surrounding water, Reproductive biology evolved in parallel with these environmental pressures. In the deep sea, individuals are widely dispersed, making encounters between males and females rare. This led to the emergence of extreme sexual dimorphism and permanent male attachment, a reproductive strategy unique among vertebrates. Larval anglerfish begin life in surface or mid-water zones, where plankton is abundant. During early development, they feed actively and grow rapidly. As juveniles mature, they descend into deeper layers, gradually adopting the morphology and physiology of adult deep-sea forms. Through this vertical life cycle, anglerfish link surface productivity with abyssal ecosystems. Over millions of years, these adaptations have allowed anglerfish to occupy one of Earth's most inhospitable habitats. Their evolutionary success demonstrates how natural selection can reshape anatomy, physiology, and behavior to match extreme environmental constraints. Today, anglerfish stand as a striking example of deep ocean specialization, revealing how life persists in darkness through efficiency, innovation, and long-term evolutionary refinement. The body of a deep-sea anglerfish reflects the extreme constraints of its environment. Every structure, every proportion, and every system is shaped by darkness, pressure, and scarcity. Unlike fast-swimming pelagic fishes, anglerfish possess soft, lightly ossified bodies. Their skeletons are reduced in density, conserving energy and allowing neutral buoyancy in cold water. Musculature is minimal, built not for speed, but for maintaining position while hovering motionless. The head dominates the body, accounting for a large proportion of total mass. This expanded cranial structure supports oversized jaws and sensory organs, and the bioluminescent apparatus, allowing the anglerfish to function as a stationary ambush predator. The jaws are loosely articulated, capable of opening to extraordinary angles. Long, transparent teeth curve inward, preventing prey from escaping once captured. Ligaments allow the mouth to expand far beyond normal dimensions, enabling ingestion of prey nearly equal to the anglerfish's own size. Behind the jaws, the stomach stretches elastically, accommodating rare large meals. This capacity is essential in environments where feeding opportunities may be separated by weeks or months. The elysium, a modified dorsal spine, extends forward from the head like a fishing rod. At its tip lies the esca, a fleshy bulb housing bioluminescent bacteria. These microorganisms produce light through chemical reactions involving luciferin and luciferase. The anglerfish regulates brightness by controlling blood flow and oxygen supply to the esca. This luminous lure serves as a visual attractant, mimicking drifting organisms in darkness, drawing small fish, crustaceans, and planktonic animals toward waiting jaws. The eyes of deep-sea anglerfish are adapted for extremely low light levels, capable of detecting faint bioluminescent flashes rather than sunlight. However, vision plays a secondary role to chemical and mechanical sensing. Sensory cells distributed across the skin detect vibration, pressure changes, and dissolved organic compounds, allowing anglerfish to perceive movement and prey presence in surrounding water without direct contact. The skin itself is darkly pigmented, absorbing nearly all incident light, rendering the body almost invisible in its environment. Some species also possess filamentous appendages that disrupt outline, enhancing camouflage. Respiration occurs through gills adapted for cold, oxygen-poor water. Metabolic rates remain extremely low, reducing oxygen demand and conserving energy. This slow physiology supports survival during prolonged fasting periods. Sexual dimorphism is extreme. Females grow large and retain predatory structures, while males remain tiny, lacking functional digestive systems in adulthood. Their sole purpose becomes locating a female upon contact. The male attaches permanently to the female's body, tissues fuse, circulatory systems merge, and the male degenerates into a sperm-producing appendage, ensuring fertilization in an environment where encounters are exceptionally rare. Together, these anatomical adaptations form a system optimized for stillness, efficiency, and survival in perpetual darkness. Every feature tuned for persistence rather than motion. In the deep ocean, where sunlight cannot penetrate, bioluminescence becomes the dominant form of illumination. Countless organisms generate light through chemical reactions, using it for communication, camouflage, or defense. But among these, anglerfish employ bioluminescence as a precise hunting tool. The esca, located at the tip of the Elysium, contains colonies of symbiotic bacteria. These microorganisms emit light continuously, powered by metabolic reactions involving luciferin and luciferase. The anglerfish regulates this glow by controlling oxygen delivery to the esca, adjusting brightness depending on environmental conditions. This light functions as a visual lure, mimicking the appearance of small drifting organisms. In complete darkness, even a faint glow attracts attention. Small fish, crustaceans, and other planktonic animals approach out of curiosity or feeding instinct. The anglerfish remains motionless, conserving energy, positioning its lure carefully in front of its mouth. When prey drifts within range, the jaws expand with sudden precision, engulfing the target in a single rapid motion. This ambush strategy minimizes energy expenditure, eliminating the need for pursuit in an environment where speed would be metabolically costly. Once captured, prey is secured by inward-facing teeth, preventing escape. The expandable stomach accommodates large meals, allowing the anglerfish to maximize rare feeding opportunities. Bioluminescence also plays a role in camouflage. Some anglerfish species use counter-illumination, matching the faint glow of downwelling light from above, reducing silhouette and making them less visible to predators below. Prey detection is not based on light alone. Mechanoreceptors sense vibration in surrounding water. Chemoreceptors detect dissolved organic compounds released by nearby organisms. These sensory systems work together, guiding feeding behavior even when visual cues are minimal. The success of this hunting strategy depends on patience rather than aggression. Anglerfish may remain stationary for hours or days, waiting for opportunity. Their slow metabolism allows prolonged stillness without significant energy loss. In this environment, every successful capture represents a substantial energetic gain stored as lipids within tissues, sustaining the fish through extended fasting periods. Through bioluminescence, anglerfish transform darkness into advantage, converting invisibility into attraction and scarcity into opportunity. This adaptation exemplifies how deep-sea predators exploit physical laws rather than oppose them, using light not to reveal themselves, but to draw life toward them. Reproduction in the deep ocean presents one of the greatest challenges faced by any organism. Individuals are scattered across vast volumes of water, population density is extremely low, and encounters between potential mates are rare for anglerfish. These conditions drove the evolution of one of the most extraordinary reproductive systems in the animal kingdom. Sexual dimorphism in deep-sea anglerfish is extreme. Females grow large, retaining full predatory anatomy, while males remain tiny, often less than 1/10 the size of females, lacking functional digestive systems once maturity is reached. Their sole biological role becomes locating a mate. Male anglerfish rely primarily on chemoreception, detecting pheromones released by females into surrounding water. These chemical signals drift with deep currents, forming invisible trails that guide males across long distances. In darkness, scent replaces sight. When a male finally encounters a female, he bites onto her body using specialized teeth. This initial attachment marks the beginning of a permanent fusion. Over time, the tissues of both individuals merge, blood vessels connect, and the male becomes physiologically integrated into the female's circulatory system. Gradually, the male's organs degenerate, except for the testes, which remain functional. He no longer feeds independently. Instead, he receives nutrients directly from the female's bloodstream, becoming a living reproductive appendage rather than a separate organism. Some females host multiple males simultaneously, ensuring continuous availability of sperm whenever eggs mature. This system guarantees reproductive success in environments where meeting even a single partner may be improbable. Fertilization occurs internally. Eggs are released in gelatinous sheets or buoyant masses that rise toward upper water layers. Larvae hatch in plankton-rich zones, feeding actively near the surface before descending into deeper habitats as they mature. This vertical life cycle connects surface productivity with abyssal ecosystems. Energy captured through photosynthesis enters deep-sea food webs through developing anglerfish larvae. Although survival rates during early life stages are low, wide dispersal increases. Genetic connectivity between distant populations, maintaining species continuity across ocean basins. This reproductive strategy represents an elegant solution to isolation, eliminating the need for repeated mate encounters and ensuring fertilization whenever conditions permit. Among vertebrates, no other group exhibits such permanent sexual fusion. Anglerfish demonstrate how evolutionary pressure can reshape reproductive biology beyond conventional limits. Through this system, Anglerfish persist in one of Earth's most inhospitable environments, sustaining populations across immense distances and geological timescales. Although adult anglerfish inhabit some of the darkest regions of the ocean, their early life begins far above, in surface and mid-water layers where plankton is abundant and sunlight still penetrates. After fertilization, females release eggs enclosed within gelatinous sheets or buoyant ribbons. These structures drift upward through the water column, carrying developing embryos toward warmer, food-rich zones. Larvae hatch as transparent, planktonic organisms, feeding actively on microscopic prey. During this phase, growth occurs rapidly, metabolic rates are higher, and sensory systems begin forming. Unlike adults, larval anglerfish possess relatively conventional fish anatomy, including functional digestive systems and more balanced body proportions. This reflects the energetic demands of surface feeding and active movement. As juveniles mature, hormonal and developmental changes initiate descent into deeper layers. Bones become lighter, muscles reduce, and the distinctive elysium begins to form. This gradual vertical migration links ocean layers biologically. Surface-derived energy captured through plankton enters deep ecosystems via descending juveniles. Throughout descent, individuals pass through mesopelagic zones populated by bioluminescent organisms and mid-water predators. Survival depends on camouflage, reduced visibility, and timing of migration. Only a small fraction reach adult depths. Many fall prey to surface predators. Others fail to locate appropriate habitats. Yet those that succeed carry genetic lineages into abyssal environments. Settlement occurs when juveniles reach chemically favorable zones along continental slopes or deep basins. From this point onward, metabolism slows dramatically. Growth becomes gradual and adult. Morphology develops fully. This life cycle strategy enables anglerfish populations to remain genetically connected across vast ocean regions. Despite adults living in extreme isolation, vertical migration also plays a role in global nutrient cycling, transferring carbon and nitrogen from surface ecosystems to deep-sea habitats. Through larval dispersal and descent, anglerfish become biological conveyors, integrating shallow productivity with abyssal persistence. This journey, from light to darkness, represents one of the most profound transitions experienced by any vertebrate, reshaping anatomy, physiology, and behavior in response to depth. Life in the deep ocean operates under constant energy limitation. Food arrives unpredictably, temperatures remain near freezing, and movement requires significant metabolic investment. Anglerfish have evolved physiological systems designed to minimize energy use while maximizing survival. Basal metabolic rates in deep-sea anglerfish are among the lowest measured in teleost fishes. Cellular respiration proceeds slowly. Enzyme activity is optimized for cold environments, and oxygen consumption remains minimal. These adaptations allow individuals to survive prolonged fasting periods that may extend for weeks or months. Muscle tissue is reduced, favoring slow, sustained contraction over rapid bursts of activity. Fins provide subtle stabilization rather than propulsion. The fish maintains position in water using minimal effort, relying on buoyancy rather than swimming. Energy storage occurs primarily as lipids within muscle and liver tissues. These reserves accumulate during successful feeding events, then support physiological maintenance during extended intervals without prey. Digestive processes also proceed slowly, maximizing nutrient extraction from each meal. Stomach contents may remain for days, ensuring complete absorption of available energy. Thermal regulation is passive. Body temperature matches surrounding water, eliminating energetic costs associated with internal heating. This ectothermic strategy further conserves resources. Circulatory systems operate efficiently at low temperatures, delivering oxygen and nutrients with minimal cardiac output. Hemoglobin exhibits high affinity for oxygen in cold water, supporting tissue function despite reduced availability. Behavior complements physiology. Anglerfish remain motionless for extended periods, reducing metabolic demand. Ambush predation replaces active pursuit. Movement occurs only when necessary. This energy-conserving lifestyle supports longevity. Although exact lifespans remain unknown, individuals are believed to survive for many years, possibly decades, within stable deep-sea environments. Survival also depends on tolerance to pressure. Cellular membranes are adapted to maintain integrity under hundreds of atmospheres. Proteins remain functional despite compression. Structural flexibility prevents damage during vertical movement. Together, these metabolic and physiological traits form a coherent survival strategy, allowing anglerfish to persist in environments characterized by cold, darkness, and scarcity. They represent one of evolution's most refined solutions to extreme resource limitation, demonstrating how life can thrive through conservation rather than consumption. In the absence of sunlight, perception in the deep ocean depends on alternative sensory channels. Anglerfish inhabit a world constructed from chemical gradients, pressure waves, and faint bioluminescent flashes. Rather than visual landscapes, their eyes are adapted for extreme low light conditions, containing large pupils and highly sensitive photoreceptor cells, capable of detecting minimal light produced by bioluminescent organisms. However, vision alone is insufficient in complete darkness, and other senses dominate daily survival. Chemoreception plays a central role. Specialized receptors distributed across the skin and within nasal cavities detect dissolved organic compounds. These chemical cues reveal the presence of prey, potential mates, and environmental changes. Scent replaces sight as the primary navigational tool. Mechanoreceptors embedded along the body sense vibration and water movement, allowing anglerfish to perceive approaching organisms without direct contact. Even subtle pressure fluctuations created by swimming prey can be detected at short distances. The lateral line system further enhances sensitivity to motion, enabling precise localization of moving targets. This system allows anglerfish to orient their bodies correctly before initiating feeding strikes. Tactile feedback contributes to spatial awareness. Fins and body surfaces register contact with surrounding water masses, providing continuous information about orientation and stability. Together, these sensory systems integrate into a cohesive perceptual network, guiding behavior without reliance on visual input. Neural processing occurs efficiently despite reduced brain size. Reflex pathways dominate, allowing rapid response to stimuli while conserving energy. Cognitive complexity is unnecessary in an environment defined by simple survival demands. These adaptations enable anglerfish to operate effectively in total darkness, locating prey, identifying mates, and maintaining position in three-dimensional space without light. They demonstrate that vision is not essential for complex behavior, and that life can construct accurate representations of its environment through chemistry and vibration alone. Even within the deep ocean, where biological activity is sparse, anglerfish exist within complex food webs, interacting with predators, prey, and microbial communities across multiple depth layers. Natural predators include larger deep-sea fishes, cephalopods, and occasionally marine mammals that dive to bathypelagic depths. Although encounters are infrequent, predation pressure persists, shaping behavior and distribution. Anglerfish reduce vulnerability through camouflage, remaining nearly invisible in darkness, minimizing movement, and relying on low metabolic demand rather than escape responses. When threatened, rapid swimming is rarely employed. Instead, individuals rely on stillness and concealment, allowing predators to pass undetected. Their primary ecological role is that of ambush predator, regulating populations of smaller fishes and crustaceans, while converting surface-derived energy into deep-sea biomass. Through feeding, anglerfish influence prey distribution and behavior, contributing to balance within mesopelagic and bathypelagic communities. They also participate in vertical nutrient transport. Larvae feeding near the surface later descend, delivering organic matter to deeper layers. Adults further redistribute carbon through respiration and waste production. Anglerfish carcasses become localized energy sources after death, supporting scavengers and microbial processes, integrating individual life cycles into broader ecosystem dynamics. Environmental threats increasingly originate from surface activities. Climate change alters plankton productivity, reducing food supply to deep waters. Ocean acidification affects physiological processes, and expanding deep-sea exploration introduces habitat disturbance. Despite these pressures, anglerfish populations demonstrate resilience rooted in low energy demand and flexible life history strategies. Though recovery from large-scale disruption would occur slowly, their presence reflects the stability of deep-sea ecosystems, serving as indicators of environmental health across abyssal habitats. Through their interactions, anglerfish connect surface productivity with deep ocean food webs, maintaining continuity between planetary systems separated by kilometers of water. The deep ocean functions as Earth's largest long-term carbon reservoir, absorbing and storing vast quantities of carbon transported from surface ecosystems. Anglerfish participate quietly in this global process through feeding, metabolism, and vertical life cycle movements. Primary production begins near the surface, where phytoplankton convert atmospheric carbon dioxide into organic matter using sunlight. This carbon enters marine food webs, eventually forming marine snow, a continuous descent of microscopic particles drifting downward through the water column. As anglerfish larvae feed in surface and mid-water layers, they incorporate this carbon into developing tissues. During descent into deeper habitats, this biomass is transferred vertically, delivering surface-derived energy into abyssal ecosystems. Adult anglerfish further process carbon through respiration, releasing dissolved carbon dioxide into deep waters, and through waste production, depositing organic material onto the seafloor. Predation by anglerfish concentrates dispersed carbon into larger biological packages. Prey consumed becomes stored energy within anglerfish bodies, later redistributed through trophic interactions or sediment deposition following death. Carcasses of anglerfish contribute to benthic carbon pools, supporting scavenger communities. And microbial metabolism, gradually becoming buried within sediments, effectively removing carbon from short-term atmospheric cycling. This biological pump operates continuously, independent of human observation, transferring carbon across thousands of meters of ocean depth. Anglerfish function as mobile components within this system, integrating surface productivity with deep ocean storage. Changes in surface plankton communities driven by climate warming, nutrient shifts, or acidification propagate downward, altering carbon availability to deep-sea organisms. Anglerfish populations respond indirectly through food supply variation. Understanding these linkages highlights how deep-sea predators influence planetary climate regulation, their slow lives intersecting with global biogeochemical cycles. Through these processes, anglerfish help maintain equilibrium between atmosphere and ocean, demonstrating that even isolated abyssal species participate in Earth-scale systems. Anglerfish populations are distributed unevenly across the world's oceans, their presence shaped by depth gradients, temperature regimes, current systems, and the slow delivery of organic material from surface waters. Most deep-sea species inhabit mesopelagic to bathypelagic zones, ranging approximately from 200 meters to more than 4,000 meters in depth. Highest densities occur along continental slopes, submarine canyons, and oceanic basins where marine snow accumulates more reliably. Adults remain solitary. Encounters between individuals are rare, except during brief reproductive interactions. Population connectivity is maintained primarily through larval dispersal rather than adult migration. Larvae transported by upper ocean currents establish genetic links between distant regions, allowing populations separated by thousands of kilometers to remain part of continuous lineages. At local scale, density is extremely low, reflecting limited food availability. Each individual occupies large three-dimensional territories, minimizing competition. Age structure within populations remains poorly understood. Growth is slow, molting intervals long, and external age markers absent. Researchers infer demographics through size distribution and reproductive condition. Environmental stability historically supported anglerfish persistence. Deep-sea habitats change gradually, allowing populations to maintain equilibrium over extended periods. However, modern climate variability introduces uncertainty into these patterns. Shifts in current circulation alter larval pathways, potentially isolating regional groups, while reduced surface productivity decreases food input to adult habitats. Despite these pressures, anglerfish remain widely distributed, demonstrating resilience rooted in flexible life history strategies and low energy requirements. Their population patterns reflect hidden ocean architecture, revealing where nutrients flow, where currents converge, and where life can persist beneath kilometers of water. For most of Earth's history, anglerfish lived beyond human reach, drifting unseen through deep pelagic waters, their existence unknown until advances in deep-sea exploration made observation possible. Early scientific encounters occurred through deep water trawling, specimens retrieved from extreme depths revealed unfamiliar anatomy, oversized jaws, glowing lures, and reproductive systems unlike any previously documented invertebrates. With the development of submersibles and remotely operated vehicles, researchers gained the ability to observe anglerfish in their natural environment, capturing footage of individuals hovering motionless, illuminating darkness with bioluminescent lures, and interacting subtly with surrounding currents. Despite technological progress, direct observation remains limited. Anglerfish occur at low densities, inhabit vast volumes of water, and avoid strong illumination. Many aspects of their behavior remain inferred rather than directly measured. Human activities now influence even these remote habitats. Climate change alters surface plankton productivity, reducing organic matter delivered to deep waters. Ocean acidification affects physiological processes, and expanding deep-sea exploration introduces noise and physical disturbance. Although commercial fishing rarely targets anglerfish directly, bycatch occurs in deep water operations. Habitat disruption affects prey availability, and chemical changes propagate downward through ocean layers. Scientific monitoring increasingly relies on acoustic surveys, environmental DNA sampling, and long-term moored instruments. These methods provide indirect insight into population trends and distribution. Anglerfish now serve as indicators of deep ocean ecosystem health. Shifts in their abundance reflect broader changes in food webs and circulation patterns. Conservation efforts emphasize precaution, recognizing that deep-sea organisms recover slowly from disturbance. Protecting deep habitats safeguards entire communities adapted to extreme environments. Through continued research, anglerfish help expand understanding of life under pressure, informing both marine biology and planetary science. Although anglerfish inhabit depths far removed from surface weather. They remain connected to planetary climate systems through ocean circulation and biological productivity. Warming surface waters alter stratification, changing nutrient upwelling and phytoplankton growth. Reduced plankton abundance leads to diminished marine snow, decreasing energy supply to deep-sea food webs. Anglerfish experience these changes indirectly through reduced prey availability. Ocean acidification introduces additional physiological stress, as increasing carbon dioxide lowers seawater pH, affecting cellular processes and potentially increasing energetic costs of maintenance and reproduction. Expansion of oxygen minimum zones compresses habitable depth ranges, forcing organisms into narrower layers, increasing competition and altering predator-prey dynamics. Changes in current systems affect larval dispersal pathways, potentially isolating populations and reducing genetic exchange across ocean basins. These shifts occur gradually, measured across decades, but long-lived species integrate impacts over entire lifespans. Anglerfish therefore provide biological records of environmental change. Predictive models suggest deep-sea ecosystems will experience delayed but persistent responses to surface climate trends. Recovery from disruption will occur slowly, emphasizing the importance of proactive management. Scientists now integrate physiological data, population monitoring, and oceanographic models to forecast future conditions. Anglerfish serve as focal species for understanding deep ocean resilience. Their fate reflects broader planetary trajectories, illustrating how atmospheric processes ultimately influence even Earth's darkest habitats. Anglerfish occupy a specialized but influential position within deep ocean ecosystems, functioning as apex or mesopredators depending on depth and region. Their feeding behavior shapes prey distribution, influencing community structure across pelagic and bathypelagic layers. By consuming smaller fishes and crustaceans, anglerfish regulate population dynamics, preventing localized overabundance and maintaining balance within sparse food webs. They also serve as prey for larger deep-sea predators, transferring energy upward through trophic levels, linking multiple biological compartments across depth gradients. Vertical life cycle migration integrates surface productivity with abyssal environments. Larvae convert planktonic carbon into biomass, which later descends to deeper layers. Anglerfish thus act as biological conveyors within the ocean's carbon pump. Their waste products contribute to particulate organic matter, enhancing microbial activity and sedimentary nutrient cycling. Even individual life histories influence benthic processes through eventual deposition of organic material. Anglerfish bodies host microbial communities, supporting bacterial metabolism and nutrient transformation, further embedding these predators within biochemical networks. Through their presence, anglerfish increase ecological connectivity, stabilizing deep-sea systems that operate under extreme energy limitation. They demonstrate how even low-density species exert cumulative influence across vast spatial scales. Subtle interactions repeated continuously shape ecosystem function. Understanding anglerfish. Ecology provides insight into the architecture of deep ocean food webs, revealing how energy flows through darkness via slow, persistent biological pathways. The evolutionary history of anglerfish reflects repeated adaptation to extreme environments. Comparative genetic studies indicate that deep-sea lineages diverged from shallow water ancestors millions of years ago, following gradual expansion into darker and colder habitats. Within the order Lophiiformes, multiple families independently evolved bioluminescent lures, demonstrating convergent evolution driven by similar ecological pressures: darkness, low prey density, and limited mobility. Morphological diversity across anglerfish species illustrates how evolutionary pathways respond to depth gradients. Shallow water relatives retain more robust skeletons and conventional reproductive systems, while deep-sea forms exhibit reduced ossification, enlarged jaws, and extreme sexual dimorphism. Permanent male attachment evolved uniquely among ceratioid anglerfish. No comparable strategy exists in other vertebrate groups, highlighting how reproductive systems can be radically reshaped when mate encounters become improbable. Comparisons with other deep-sea predators, such as gulper eels, viperfish, and dragonfish, reveal shared traits including expandable stomachs, oversized teeth, and slow metabolic rates. Yet anglerfish remain distinct in their use of bioluminescent lures and reproductive fusion. Genomic analyses suggest that pressure tolerance in anglerfish arises from modifications to membrane proteins and enzyme structures, allowing cellular processes to remain stable under hundreds of atmospheres. Cold adaptation involves altered lipid composition in cell membranes, preserving flexibility at low temperatures, while metabolic enzymes exhibit temperature-dependent efficiency optimized for near-freezing conditions. These physiological traits mirror those found in other abyssal species, illustrating parallel solutions to shared environmental constraints. Anglerfish therefore serve as model organisms for studying deep-sea evolution, providing insight into how vertebrate biology adapts to darkness, pressure, and scarcity over geological time. Their evolutionary narrative demonstrates that extreme habitats do not merely limit life, they reshape it, generating forms and strategies unseen elsewhere on Earth. For much of their evolutionary history, anglerfish inhabited environments protected by depth and distance. Human influence faded long before reaching bathypelagic waters, yet modern technology increasingly bridges this separation, introducing new pressures into deep ocean ecosystems. Although anglerfish are not directly targeted by commercial fisheries, deep water trawling results in bycatch. Habitat disruption alters prey availability, and sediment disturbance affects chemical gradients essential for deep-sea life. Emerging industries such as deep-sea mining pose additional risks. Extraction of mineral resources resuspends fine sediments, reduces water clarity, and disrupts benthic communities across wide regions. Anglerfish populations may be affected indirectly through food web destabilization. Climate-driven changes compound these threats. Altered surface productivity reduces marine snow, limiting energy supply to deep habitats. Acidification increases physiological stress, and expanding oxygen minimum zones compress habitable depth ranges. Deep-sea organisms recover slowly from disturbance. Long lifespans, delayed maturity, and low reproductive output limit rapid population rebound. Anglerfish exemplify this vulnerability. Conservation strategies increasingly emphasize precautionary approaches, establishing protected deep-sea regions, regulating industrial activity, and monitoring ecosystem health through indicator species such as anglerfish. Scientific research continues to refine understanding of population dynamics, genetic connectivity, and physiological tolerance thresholds. These data inform policy decisions aimed at preserving deep ocean biodiversity. Public awareness of deep-sea life grows through exploration, imagery, and education, transforming abstract depths into recognizable ecosystems deserving protection. The future of anglerfish depends on responsible stewardship of ocean resources. Actions taken at the surface will determine conditions in darkness far below. Anglerfish offer a unique perspective on how vertebrate life adapts to extreme environments. Their biology reflects long-term refinement under conditions of darkness, pressure, and limited energy availability. From their anatomy, we learn that efficiency can replace strength. Soft bodies conserve resources. Expandable jaws maximize rare feeding opportunities, and reduced musculature supports prolonged stillness. From their metabolism, we observe how slow physiological processes extend survival. Minimal oxygen consumption, lipid-based energy storage, and passive thermal regulation together sustain life across extended fasting periods. From their sensory systems, we see that perception does not depend solely on vision, chemical detection, vibration sensing, and lateral line input provides sufficient environmental awareness in total. Darkness. Their reproductive strategy illustrates how evolution solves isolation. Permanent male attachment eliminates the need for repeated mate encounters, ensuring fertilization in sparsely populated habitats. Their life cycle demonstrates vertical integration of ocean layers. Larvae feed in productive surface waters. Adults persist in abyssal depths. Energy and genetic material move continuously between these zones. Anglerfish embody principles of ecological balance. Survival through restraint, persistence through adaptation, and integration rather than dominance within their environment. They reveal that complexity can arise from scarcity, and that stable ecosystems do not require abundance, only continuity. These lessons extend beyond marine biology, offering insight into planetary resilience, climate regulation, and the capacity of life to occupy nearly every available niche. As scientific instruments withdraw and submersible lights fade, anglerfish resume their quiet existence within deep pelagic waters, hovering motionless in cold darkness. guided by chemistry and subtle currents rather than visual cues. Their bioluminescent lures continue to glow softly, illuminating only centimeters of surrounding water. Prey drifts past unaware, and feeding occurs without disturbance. Metabolic processes return to baseline. Digestion proceeds slowly, and energy reserves rebuild after successful captures. Each individual maintains position within vast three-dimensional territories. Larvae already dispersed elsewhere drift through upper layers, descending gradually toward deeper zones, ensuring population continuity across ocean basins. Above, surface conditions fluctuate, storms pass, temperatures rise and fall. Yet below, environmental change arrives slowly, buffered by immense water columns. Anglerfish remain embedded within these deep rhythms, participating in nutrient cycling, carbon transport, and food web regulation without awareness of surface dynamics. Eventually, individuals complete lifespans measured in years or decades, their bodies becoming localized energy sources for scavengers and microbes, reintegrating biological material into benthic systems. No traces persist beyond redistributed nutrients and altered microbial activity. Continuity is maintained through larvae rather than monuments. Returning to the deep is not disappearance. It is reintegration into planetary circulation. Anglerfish persist quietly beneath kilometers of water, carrying gentle light through darkness, sustaining ecosystems through slow and continuous biological processes. Anglerfish represent one of the most specialized evolutionary responses to life in the deep ocean. Their anatomy, physiology, and behavior illustrate how vertebrate organisms adapt to extreme pressure, perpetual darkness, and chronic energy limitation across millions of years. Natural selection refined traits that prioritize efficiency over speed, conservation over expansion, and integration over dominance. Soft bodies reduce energetic cost. Expandable jaws maximize rare feeding events. Bioluminescent lures convert invisibility into attraction, and permanent reproductive attachment resolves isolation. Through their life cycle, anglerfish link surface productivity with abyssal ecosystems. Larvae convert planktonic carbon into biomass. Adults redistribute this energy through predation, respiration, and waste production, contributing to long-term carbon storage in deep waters. Their ecological role extends beyond predation. Anglerfish influence prey populations, support microbial processes after death, and participate in global biogeochemical cycles that regulate Earth's climate. Modern exploration has only begun to reveal their biology. Vast regions of their habitat remain unobserved, and many species are known from only a handful of specimens. Continued research will refine understanding of population dynamics, physiological limits, and responses to environmental change. As human activity increasingly reaches into deep waters, anglerfish serve as indicators of ecosystem stability. Their persistence reflects the health of pelagic food webs and deep ocean circulation. Protecting deep-sea environments preserves not only individual species, but planetary systems that operate beyond direct human perception. Anglerfish remind us that much of Earth's life exists in darkness, sustained by slow processes, shaped by gravity and chemistry, and governed by continuity rather than spectacle. Their gentle lights continue drifting through abyssal waters, illuminating only moments, yet participating in cycles that span oceans and centuries. At depths exceeding 1,000 meters, hydrostatic pressure rises beyond 100 atmospheres, compressing tissues, altering protein structure, and challenging cellular stability. Anglerfish persist here through finely tuned genetic adaptations. Comparative genomic studies reveal modifications in membrane-associated proteins, preserving flexibility under pressure. Lipid compositions within cell membranes shift toward unsaturated forms, preventing rigidification in cold, compressed water. Enzymes involved in metabolism exhibit altered folding properties, maintaining functionality despite pressure-induced distortion. These molecular adjustments allow biochemical reactions to proceed efficiently where ordinary proteins would fail. Heat shock proteins and molecular chaperones remain active, stabilizing cellular components, repairing misfolded proteins, and protecting tissues from pressure-related stress. Genes regulating oxidative metabolism are downregulated, reducing reactive oxygen species production, limiting cellular damage during prolonged low oxygen exposure. DNA repair pathways show enhanced activity, countering pressure-induced molecular instability, supporting long-term survival across extended lifespans. These adaptations illustrate how selection operates not only on visible anatomy, but at microscopic molecular levels, reshaping internal systems to match abyssal constraints. Anglerfish genomes therefore represent evolutionary blueprints for life under compression, revealing how vertebrates can occupy environments once thought biologically inaccessible. The bioluminescent lure of the anglerfish represents one of the most refined examples of symbiosis in the deep ocean. Its soft, glowing esca is not self-luminous. Instead, it houses dense populations of specialized bacteria that produce light through biochemical reactions. These bacteria belong primarily to genera such as Photobacterium and Vibrio, organisms capable of emitting continuous light when supplied with oxygen and nutrients. Anglerfish. Embryos hatch without luminous bacteria. Colonization occurs later through seawater exposure or maternal transmission. Once established, bacterial populations are maintained within protected crypts inside the esca. The fish regulates light intensity by controlling oxygen delivery to these chambers. Increased blood flow enhances bacterial metabolism, producing brighter glow. Reduced flow dims the signal. Nutrients supplied by the anglerfish sustain bacterial populations, while the bacteria provide illumination in return. A mutually beneficial relationship refined through millions of years of coevolution. Genetic sequencing reveals that these symbionts have lost many free-living traits, becoming specialized for life within the lure, unable to survive independently for extended periods. The spectral quality of emitted light matches wavelengths most visible to deep-sea organisms, maximizing effectiveness in prey attraction. Some species modify the shape and movement of their lure, creating rhythmic patterns that mimic drifting prey, further enhancing hunting success. This microbial partnership converts chemical energy into biological light, enabling anglerfish to manipulate their environment in complete darkness. It demonstrates how evolution integrates microorganisms into vertebrate physiology, producing composite organisms whose survival depends on collaboration across kingdoms of life. Predation in the deep ocean follows principles distinct from surface ecosystems. Prey density is low, movement is energetically expensive, and visibility is minimal. Anglerfish exemplify one strategy among several evolved by abyssal hunters, while anglerfish rely on bioluminescent attraction and ambush. Other deep-sea predators employ alternative methods. Viperfish use elongated teeth and rapid strikes. Dragonfish utilize photophores to illuminate prey. Gulper eels engulf targets through extreme jaw expansion. Despite these differences, common themes emerge: oversized mouth, expandable stomachs, slow metabolic rates, and reliance on sensory detection rather than pursuit. Anglerfish remain unique in combining light-based attraction with complete stillness, minimizing energy expenditure while maximizing capture probability. Some predators actively patrol water columns, others drift passively. Anglerfish occupy a stationary niche, converting darkness into opportunity through microbial illumination. These contrasting strategies partition limited resources across depth layers, reducing direct competition, allowing multiple predators to coexist within sparse environments. Comparative analysis reveals how evolution explores diverse solutions to identical constraints, each species refining specialized tools for survival in darkness. Anglerfish demonstrate that successful predation need not involve speed or strength, only precision, patience, and ecological alignment. The transition from larval anglerfish to deep-sea adult represents one of the most dramatic morphological transformations among vertebrates. This process unfolds gradually as individuals descend from sunlit surface waters into abyssal darkness. Larvae hatch with relatively symmetrical bodies, functional digestive systems, and conventional fin structures. During early development, growth is rapid, fueled by abundant planktonic food. As juveniles migrate downward, hormonal signaling initiates structural reorganization. Bones reduce in density, muscles decrease in mass, and the elysium begins forming above the head. Pigmentation increases, skin darkens, and sensory systems reorganize, favoring chemoreception and mechanorception over vision. Jaw architecture expands progressively, ligaments loosen, teeth elongate, and stomach elasticity increases, preparing individuals for infrequent but large feeding events. Neural circuitry adapts to new sensory priorities. Reflex pathways strengthen. Behavioral responses shift from active swimming to stationary hovering. Reproductive tissues differentiate according to sex. Females retain full predatory capacity. Males reduce digestive function and develop attachment structures. These changes occur across multiple developmental stages, synchronized with depth transitions, illustrating how environment directly shapes anatomy through regulated gene expression. Development in anglerfish exemplifies phenotypic plasticity guided by ecological context. Surface life supports growth. Deep life selects efficiency. This transformation enables anglerfish to occupy ecological niches inaccessible to most vertebrates, completing one of nature's most remarkable life history transitions. In the deep ocean, survival does not require complex cognition. It requires efficient response to limited stimuli. Anglerfish exhibit streamlined neural architecture optimized for reflex-based behavior rather than advanced learning. Brain size relative to body mass is reduced compared to many shallow water fishes. Energy investment favors sensory integration and motor coordination over higher-order processing. Neural pathways prioritize rapid transmission from sensory receptors to feeding musculature, minimizing reaction time once prey enters capture range. Behavioral repertoires are simple: hovering, lure, positioning, jaw expansion, and reproductive attachment dominate daily activity. Unnecessary actions are eliminated through evolutionary refinement. Memory and learning appear minimal. Environmental predictability at depth reduces need for behavioral flexibility. Instead, genetically encoded responses govern most interactions. Autonomic regulation maintains buoyancy, respiration, and circulation without conscious control, conserving metabolic resources. This neurologic economy reflects environmental stability, where few variables change rapidly, and survival depends on patience rather than adaptation to novelty. Anglerfish therefore illustrate how neural systems evolve in response to ecological demands. Complexity is retained only where functionally necessary. Beneath visible predator-prey interactions, deep-sea ecosystems are sustained by microbial networks that recycle organic matter and regulate nutrient availability. Anglerfish participate indirectly in these invisible systems. Through feeding and waste production, anglerfish release dissolved organic compounds into surrounding water. These substances fuel bacterial metabolism, supporting microbial communities that form the base of deep ocean biochemical cycles. Fecal pellets and partially digested material descend slowly, becoming substrates for heterotrophic bacteria. These microbes convert complex organic molecules into simpler compounds, making nutrients accessible to other organisms. After death, anglerfish bodies become concentrated microbial habitats. Scavengers remove soft tissue, while bacteria colonize remaining material, accelerating decomposition and sediment incorporation. Bioluminescent symbionts within the ESCA represent another microbial partnership, illustrating how anglerfish integrate bacteria into physiology for functional advantage. Together, these interactions link vertebrate predators with microscopic life, demonstrating that deep-sea food webs extend beyond visible organisms into chemical and microbial domains. Anglerfish therefore occupy multiple ecological roles simultaneously: predator, nutrient redistributor, and microbial substrate provider. Their existence reinforces the concept that deep ocean ecosystems depend on continuous cooperation between macrofauna and microorganisms. Precise lifespans of deep-sea anglerfish remain difficult to measure. Direct long-term observation is limited, and traditional aging markers such as otolith growth rings are challenging to interpret under abyssal conditions. However, physiological indicators suggest that many species live for extended periods, possibly decades. Slow metabolism, low activity levels, and reduced oxidative stress contribute to prolonged cellular stability. Growth occurs gradually. Energy investment prioritizes maintenance over rapid expansion. Reproduction is episodic rather than continuous, reflecting constrained resource availability. Oxidative damage accumulates slowly, supported by efficient antioxidant systems and reduced metabolic output. These traits delay senescence and support long-lived survival. Life history strategies emphasize persistence rather than rapid turnover. Few offspring reach adulthood, but those that survive may contribute genetically for many years. This approach contrasts sharply with surface fishes that mature quickly and reproduce frequently. Anglerfish instead embody longevity under scarcity. Their aging processes illustrate how reduced metabolic demand correlates with extended lifespan, offering insights into vertebrate longevity mechanisms. Although the deep ocean appears silent to human observers, it is permeated by complex acoustic environments, low-frequency currents, distant tectonic activity, biological movement, and increasingly anthropogenic noise. Anglerfish inhabit these soundscapes without reliance on auditory communication, lacking specialized hearing structures seen in some pelagic fishes. Instead, vibration-sensitive mechanoreceptors detect pressure waves traveling through water. These receptors allow anglerfish to sense approaching organisms, shifting currents, and environmental disturbances, providing spatial awareness in darkness. Natural acoustic signals include sediment movement, seismic tremors, and biotic sounds generated by crustaceans and fishes. These form consistent background patterns to which anglerfish physiology is adapted. Human-generated noise from shipping, seismic surveys, and deep-sea exploration introduces novel acoustic disturbances, altering vibration fields that anglerfish use for orientation. Although behavioral impacts remain under investigation, increased noise may interfere with prey detection and spatial stability, particularly for organisms relying on subtle mechanosensory input. Anglerfish responses appear minimal at short timescales, yet long-term exposure could influence feeding efficiency and distribution. Understanding deep-sea acoustic ecology is essential for evaluating anthropogenic impacts on abyssal species. Anglerfish serve as models for vibration-dependent navigation systems. Fish inhabiting different ocean depths exhibit distinct physiological strategies. Surface species prioritize speed, visual acuity, and rapid metabolism. while deep-sea organisms emphasize conservation, sensory efficiency, and pressure tolerance. Anglerfish exemplify this abyssal model. Their oxygen consumption rates are dramatically lower than those of epipelagic fishes. Cardiac output remains minimal, and muscle fiber composition favors endurance over burst performance. Blood chemistry is adapted for cold, high-pressure environments. Hemoglobin exhibits enhanced oxygen-binding affinity, supporting tissue function in low-oxygen conditions. Cell membranes contain elevated levels of unsaturated fatty acids, maintaining flexibility under compression and cold temperatures. Enzyme systems demonstrate pressure-resistant folding patterns. Digestive enzymes operate slowly but efficiently, maximizing nutrient extraction from infrequent meals. Comparisons with mid-water predators reveal gradual physiological transitions across depth gradients. Metabolic rate decreases, skeletal density lightens, and sensory reliance shifts from vision to chemical and mechanical input. These patterns illustrate how environmental pressure reshapes vertebrate biology systematically. Anglerfish represent one endpoint along this continuum. Their physiology provides insight into limits of vertebrate adaptation, revealing how life extends into regions once considered biologically inaccessible. Anglerfish represent one of the most comprehensive examples of vertebrate adaptation to extreme environments. Their biology integrates anatomical innovation, physiological efficiency, microbial symbiosis, and reproductive specialization into a single coherent survival system. Across evolutionary timescales, natural selection refined traits that prioritize conservation over motion, patience over pursuit, and integration over independence. Soft bodies reduce energetic cost. Expandable jaws maximize rare feeding events. Bioluminescent lures convert darkness into opportunity, and permanent reproductive attachment resolves isolation. From genetic modifications supporting pressure tolerance to metabolic pathways optimized for scarcity, every level of organization reflects alignment with abyssal constraints. Anglerfish also function as connectors within ocean systems, transferring surface-derived energy into deep ecosystems, participating in carbon sequestration, supporting microbial networks, and shaping prey dynamics across depth layers. Their life histories illustrate how vertical migration links epipelagic productivity with bathypelagic persistence. Larvae convey nutrients downward, adults redistribute energy laterally, and decomposition returns organic material to sediments. Modern research continues to uncover previously unknown dimensions of anglerfish ecology. Yet vast regions of their habitat remain unexplored. Each expedition reveals new species, new behaviors, and new physiological strategies. As climate change and industrial expansion increasingly affect deep ocean environments, anglerfish serve as indicators of ecosystem integrity. Their resilience reflects system stability. Their decline would signal broader disruption. Protecting deep-sea habitats preserves not only anglerfish populations, but planetary processes that regulate climate and nutrient cycling. Anglerfish remind us that much of Earth's life unfolds beyond human perception, sustained by slow rhythms, subtle chemistry, and continuous biological exchange. Their quiet lights persist beneath kilometers of water, illuminating only moments, yet contributing to cycles spanning oceans and centuries.